As an advocate ng biotech na hindi ako nahihiyang i-share yung aking experience at ako ay natutuwa na ma-share yung aking experience, kailangan natin talaga na ma-involve ang agriculture sa technology. Kung wala tayong technology, maiiwan po ang Pilipinas. Ako po si Rosalie Eliasus, taga Barangay Santa Maria, San Jacinto, Pangasinan. Isa akong farmer ng rice and corn. Pero at the same time, isa akong government employee bilang Municipal Disaster Risk Reduction ng LGU San Jacinto and at the same time, the OIC-SB Secretary. Ako po ay isang medical technologist grad actually. But because after na nag-OFW, umuwi ako ng Pilipinas at hindi ko alam yung gagawin ko. But bilang IPM FFS student ng government, doon ko napag-aralan ang pagsasaka both on rice and corn. Then it was in the year 2002 nung ma-involve ako sa corn IPM FFS. Pagka-graduate ko doon sa conventional corn, nagkaroon po ng field trial sa Santa Maria, Pangasinan at doon ko nakita at naobserbahan yung pagdating ng BT corn dito sa Pangasinan. So nakita namin yung mga members ng Farmers Federation ng San Jacinto, talagang nakita namin ang pagkakaiba ng BT and non-BT. At timing na 2003, maaaprubahan na pala ang BT corn dito sa Pilipinas. And from that on, not only because I started BT corn dito sa field na ito, but at the same time as the pioneer and the role model ng BT corn. Pagka conventional ka, meron talaga yung timing from the time na magplaplowing ka na maraming ilang beses. And then, not only on the preparation, but time of fertilization, time kung kailan ka magwi-weeding, time kung kailan ka noon ang uso sa amin is detasseling, o kaya mag-i-spray ka kasi yun yung pinag-aaralan mo sa IPM FFS. With that, pati yung kung kailan ka magpapatubig, marami. At sa kung kailan ka mag-weeding, kasi maraming weeds na tumutubo. Kailangan mo pa rin na mag-uupa ka ng mga tao. So it's laborious. But with the BT corn, parang shortcut ang nangyari. Pag nakapagtanim ka na kaagad ng corn, maghintay ka lang ng at least 20 days. After 20 days, because we have 2 traits now, nag-i-spray na lang kami ng glyphosate. O di wala ng weeds at that time. Bumababa ang iyong production cost. At the same time, ang maganda is naging high yield ang BT corn. Hindi na rin nare-reject ng mga feed millers, yung mga traders. Kasi ang problema din noon ng conventional, nagkakaroon kasi ng aflatoxin. But this time, because there is no infestation, kaya wala siyang aflatoxin. So, yun yung mga differences. At pag high yield ka, di mas maganda ang profit mo. Nakita ng mga group ng farmers dito sa San Jacinto. Kaya agad-agad din silang naging adopter ng ating BT corn. And on that time, marami akong nahikayat kasi naikwento ko rin sa ating mga iba-ibang meetings, naiimbitahan ako sa mga ibang lugar na nagmamais. So, doon nag-start na parang, kaya nga nabansagan ako ng Queen of BT corn kasi ako yung nauna na naglalaganap sa mga iba-ibang towns dito.
Ako po si Sosimo Gonzales, 70 years old, taga Kasibong, San Jacinto, Pangasinan po. Sa pagtatanim po ng BT corn, ay ang nag-introduce po sa amin yan ay sila Madam Rosalie. Tapos, nung masubukan namin, maganda naman po ang naging resulta. High yielding yung BT corn at hindi kami masyado namumroblema sa corn borer. Sa BT corn po, less expenses sa insecticides at high yielding naman po yung BT corn. Maganda silang umani. Kung ikukumpara doon sa conventional na mais. Minimal lang ang magagamit gawa ng hindi na mako-corn borer. Although, pag later stage, meron po namang ding leafhoppers. Pero manageable naman. Tayong mga magsasaka minsan, kailangan maniwala din tayo sa mga modern technology na ginagamit. Hindi naman po niyan i-introduce kung makakapag-down sa mga farmer eh. Kaya doon ako naniwala. Itong late harvest ko na 1.7 hectares, naka 15 tons po ako ng 15 tonelada po. Proven naman na malaki ang kita at may bintahe siya kumpara doon sa conventional na mais.
Ako po si Romeo Velasco, nickname Romy, taga Santa Maria, San Jacinto, Pangasinan. Mas maganda ang gem product. Ang Biticorn, may pagbabago sa farming natin kasi ang kagandahan nito, bawas trabaho, bawas gastos, dagdag kita. Walang-wala ako. Sa malit pala yung mga tinataniman ko, bukid lang ng daddy ko. Kasi kakunti naman, malit naman yung taniman namin noon. Hanggang nag-upisa ako nagtanim ng Biticorn, hanggang after 2 years, nag-expand ako ng 5 hectares. And then, lumaki na naman ang kita, nag-expand ako na naman ako ng 5 hectares, maging 10 hectares, hanggang lalo akong kumikita. Doon ako nag-expand ng, ngayon, itong taon na to, nag-expand ako ng 25 hectares. Sa kita lang sa Biticorn. Ang talagang pangarap ko para sa akin, matulungan yung mga farmers na maliliit para maingganyo silang magtanim ng Biticorn. Kikita din sila ng malaki at makakatulong din sa mga pamilya nila. At saka sa akin pa, gusto ko mag-expand ng taniman para makatulong din ako doon sa mga katuwang kong kasamahan na trabador para matulungan ko rin din sa kanilang pamilya at makapagpaaral pa sila ng mga anak nila at saka pagkain nilang araw-araw. Kung talagang ikarir mo talaga nasa puso mo pagtatanim ng mais na Biticorn, talagang kikita ka.
Ako si Trinidad Velasco, taga Barangay Imelda, San Jacinto, Pangasinan.
Ako rin si Saturnino Velasco Senior.
Talagang certified farmer kami. Ang farmer talaga, pinapakain niya yung ang dami, 5,000 Pilipino. Simula nung nagtanim kami ng Biticorn, hindi namin naranasan na naapektuhan yung mais namin doon sa corn borer. Kaya masaya kami na bilang farmer. Siyempre, tungtuwa kami kasi yung production namin ay tumaas. Tapos, dahil sa produkto ng lupa namin, yun, nakatapos yung anak namin na 5. Mayroong 2 nurses, tapos 2 engineer, tapos isang babae na dentist. Nung una, ordinary mais pa tinatanim namin. Ngayon po, pagkatapos a year, yung ordinary, kakaunti yung produkto. Ngayon, pinalitan namin yung Biticorn. Nakita namin, nung tinanim namin, higher production talaga.
Nakita ko na doon sa trial na ginawa namin. At the same time, ginawa namin nung demo trial dito actually is nung graduation namin, nung field day namin after 120 days. Nagtira ho kami, sir, ng talagang binoil namin. Ang ginawa namin is nilaga namin yung BT at saka yung non-BT. Naglaga kami ng mais para kami mismong mga farmers titikman namin. Wala naman nangyari sa amin na sabi nila ay magkakasakit ka o sasakit ang tiyan mo, mag-allergic ka. All those things narinig na po namin yun. But so far wala namang nangyaring ganun. Until now, I'm 62 years old but look, wala namang nangyari hanggang ngayon. Yun na rin po yung pinapakain namin sa baboy at sa mga manok. Ginagawa na ng mga kasama ko, yung mga malapit ng i-harvest, kinukuha na, hindi na nakakahintay. Yun na yung pinapakain sa mga pato at manok. So there's nothing to fear. Meron akong mga laborers, mga farm workers na mini-maintain ko rin po yung kanilang pangkabuhayan dahil may mga anak na rin po sila, may pamilya. At hindi ko binitawan ang farming because of these people. At ngayon, yun, napagawa ko na rin yung shop doon sa labas. Unti-unti, acquire na rin po ng mga ibang bagay na maski papano convenient naman sa buhay. Kailangan natin talaga na ma-involve ang agriculture sa technology. We are still looking forward for other technologies, not only on these two traits na meron tayo ngayon, glyphosate and the Bacillus thuringiensis. Palagi kong binabanggit pagka nagkakausap kami sa mga scientists ay the drought resistant or even the water resistant. Kasi ang weather is hindi natin alam. Maya-maya, tingnan mo, kanina umulan. Bukas, sobrang araw. So those are the things na kailangan natin pang pagtuunan ng pansin ang other technologies for not only on corn but other crops probably.
Alright, good afternoon. Magandang hapon po sa ating lahat. Are we all ready for this afternoon seminar? Hello, are we all ready for this afternoon seminar? Yes, so we also have some participants joining us via Zoom and Facebook. So we have over 30 participants here, warm bodies here, and we also have over 100 participants via Facebook and Zoom. So magandang hapon po sa ating lahat. Good afternoon. This is the Agriculture and Development Seminar Series, or ADSS, brought to you by SEARCA. And this time, we are very happy to partner with the International Service for the Acquisition of Agri-Biotech Applications, or ISAAA, and the United States Department of Agriculture Foreign Agricultural Service, Manila. My name is Sunny, and I'll be your MC for this afternoon. This is ADSS on breeding innovations for increasing productivity and climate resilience in crops and livestock. So for almost 20 years now, the ADSS has been facilitating a vibrant exchange of scientific and practical knowledge in agriculture and development within and even beyond the Los Baños science community. So we're happy to be joined this afternoon by some students. Do we have students here? Okay, some students, some faculty and staff from the Los Baños science community, perhaps from the OST, also from IPB. And joining us as well online, we also have a diverse set of attendees. So some house rules before we begin. We know the drill. So if you're on Zoom, so please mute your microphones, position your camera properly because we're gonna have some group photos later. And for on-site participants, of course, please keep your phones in silent mode and no eating and drinking also. All right. So for any social media posts, please, we're being streamed live on Facebook through the SEARCA Facebook page. You can use the hashtag SEARCAADSS. Malay ninyo, mamaya may premyo ang may post. Okay. So to formally begin and welcome us all, I'd like to call on the Deputy Director for Programs of SEARCA, Dr. Nur Azura Adam.
Thank you, Sunny. Thank you very much, our MC, for this afternoon. And first of all, welcome to all to SEARCA. I think most of them are students from UPLB, I guess. Yeah, all right. So first of all, Mr. Mark Hansel, we meet again. And we also have our Executive Director, Dr. Rhodora Romero Aldemita, or Ma'am Ola, yeah, and also Professor Hugh Jones, that is, he's here with us online. Dr. Claro, Mingala, so they are all our resource speakers for today. Friends, students, staffs of SEARCA, ladies and gentlemen, again, good afternoon, everyone, and welcome to the Drillon Hall, SEARCA. For your information, this is the final series of our biotechnology outreach activities. Yeah? So although this is a final one, doesn't mean that it stopped here. I'm sure that there will be also other engagement in different kind of activities that we will always try to educate the people about biotechnology in the true manner. Okay, ladies and gentlemen, the first two sessions that we have before, we have an engagement with one of the province, which is in Nueva Ecija, and also we have another engagement with the members of judiciary, and I have the opportunity to be in the third series in the House of Representatives, where it is a very, very engaging and fruitful discussion, and this will be the fourth one. So let us take this opportunity for those who have, who is the first time to be here in the biotechnology activity outreach series. So I hope you will gain some knowledge, because we'll try our best to give, to educate people the right way about biotechnology. Nobody, we always have the negative kind of thing about biotechnology, and I hope that this will open up our mind. And of course, I also would like to, when I see the videos just now, maybe in future we would like to invite maybe the farmers to be the speakers, to show, to even convince, sometimes to convince other farmers, it makes another farmers to do it also, right? So I guess that that will be, maybe we can plan in the future. So enjoy, everyone. Make it not, I know it's in the afternoon, after lunch. So just bear in mind, because we have a very, very good resource person today to share with you and to be engaged with you. Just make sure that if you have any questions, you have any doubts, please, at the end of the presentation from both our from all our speakers, you can we can go for the Q&A session. With that, ladies and gentlemen, have a pleasant session this afternoon. Thank you very much.
Thank you, Dr. Noor. Now we would also like to hear from the Executive Director of ISAAA and the Director of the Global Knowledge Center in Biotechnology. We have with us this afternoon Dr. Rhodora Romero Aldemita.
Good afternoon, everyone. Yes, I reiterate what Dr. Ahmad had said. It's afternoon, and I hope you're not going to doze off. But I'm very happy that we have more online participants. Not really happy, but surprised. I was really thinking that this hall will be full with a little bit online, but it's the reverse. But it's okay. We're not going to give them merienda. You will be having merienda later, so it's a bit different. So good afternoon again, and welcome. This is the fourth one that we're having, as Dr. Ahmad said. This is going to be for Biotechnology Forum on Breeding Innovations for Increasing Crop Productivity and Climate Resilience in Crops and Livestock. This is a bit different from what we have done from the previous ones, and this is actually geared towards the technology developers, the upcoming technology developers, which are our students. So we would like to focus more on how we would entice our students to go for biotechnology, because we're really needing new ideas, new people, and a lot of us are already retiring. And so we'd really like to invite most of our students to go for agri-biotechnology in their career, and we need regulators as well, and communicators, science communication, and hopefully after this you would have already some idea on what you want to do when you grow up. Did you grow up? Okay, so yes, so today we're going to have 3 speakers. The first one would be yours truly. I will be talking about crop biotechnology, the ones in the pipeline, the benefits. It has been there since 1996 up to 2020. And then we have Dr. Mingala, who will talk about livestock. And then we have our online speaker, Dr. Hugh Jones, who will talk about what's happening with new breeding innovations and the regulation that is prevailing, the current discussion on the regulations of new breeding innovations. So also we would like to focus on the upcoming ones, especially crops and livestock. And we are thinking that we have already covered all of the stakeholders, because the last time that we had it, it was in Muñoz, We have the ISAAA, and of course, the technology developers, everybody there in the science community of Muñoz, from Region 1 to Region 3. Now we are having Region 4 and downwards, but we'd like to continue more. This is not going to be the end. Actually, this is the fourth, but we have already gone through with the judiciary and the House of Representatives, but we need some more. So that's why we're not stopping here, you're correct. And we hope that whatever learnings that you will have from this discussion, you will share it with your friends, with your co-students, with your organizations. We're happy that there are student organizations represented here. And so with that, thank you very much for coming, and happy learning to everybody.
Okay, happy learning. So later we're going to give out some prize for anyone who would be posting about their insights or key takeaways from this. And don't forget to use the hashtag SEARCA ADSS, because we're going to look for that post and we're going to give a prize randomly to anyone who would be posting about it. All right. Now, finally, we are also lucky to have with us this afternoon the Agricultural Attaché of the USDA FAS of the U.S. Embassy Manila to give us a short message. We have Mr. Mark Hansel.
Thank you so much, Sunny. And Dr. Adam, Dr. Aldemita, Dr. Mingala, all of our good friends involved in the biotechnology efforts here. It's an honor to be with you today and to represent the United States Department of Agriculture and the United States Embassy. I'm Mark Hansel, Agricultural Attaché at the U.S. Embassy, and it is my privilege to be serving in the Philippines right now and to have a chance to connect with Philippine agriculture and to help find areas where we can work together and help solve the challenges of feeding the world. That is really what my agency is about. We are a trade agency, but as a trade agency, our efforts are all about taking things that are produced in abundance and making sure they get to where they need to be, where the greatest needs are. And trade is an indispensable component of food security, but so is embracing technologies that allow us to produce foods in better, more efficient ways, and in ways that fit the places where we are. Dr. Mingala is with his work with the Philippine Carabao Center, doing a lot of interesting and important things there in terms of developing a species that is that's been in the Philippines since how many centuries? I don't know, but are they native? Okay, all right, so who knows? But that's why carabao, they can thrive here. Other species of cattle, there have been some challenges. Biotechnology holds a lot of promise. I've said this before at other events, but I'm from the great state of Idaho in the northwestern U.S., and when I go out and about in the Philippines and I see Brahman cattle, I think of rodeos, because the bull riding is always Brahman cattle that they use for the bulls. And here in the Philippines, you see Brahman cattle, and one of the biggest reasons that those cattle have been brought to the Philippines is that the climate works for them here. But it still is not meeting the challenge of the milk demand, the dairy demand of the Philippines. So there are a lot of things that biotechnology holds promise. Heat-resistant strains of animals, climate change-resistant plants. There are so many different things you can do with this. And let me tell you a little disclosure right now. I think pretty much everybody in this room, each person in this room, understands the science of biotechnology better than I do. I am a trade officer, and by definition, my job is that of a generalist, to help bring the U.S. policies in here and to help us align things and see where we can bring resources to help with these efforts. And so I'm thrilled to see all of you here today, especially because so many of you, all these young faces that I'm seeing there, represent the future of agriculture, represent the future of agricultural innovation in the Philippines, and you're going to be doing some really important work in the coming years. And some of the most important things you will do will happen in spaces like this because people need to understand, or they will not embrace the technologies and the benefits of this. A lot of what is in the way of biotechnology in agriculture is simply misunderstanding. It is misinformation. And when people truly understand the science, they understand how it works, they understand that we are talking about a lot of techniques and things that have been tested for decades and proven safe and effective, and yet still people we still see policy sometimes succumbing to the pressures of misinformed views. And so it's really important that we do the things that we're doing here today. It's really important that we take the knowledge and we go out there with it and we help people understand these things better. And I also want to say, and Sunny's been talking about some of the more procedural aspects of this, that as far as USDA is concerned, we appreciate everybody who's attending online and we extend them a big welcome. But those of you who are here in person, some of you will be really glad you came, because even Florence, my colleague here, who is our biotechnology specialist in our office, we brought down a few prizes that will be going to people who ask questions. So please, in life, curiosity is its own reward, but it's okay sometimes if that is augmented with something tangible. So we thank you for coming. We wish this event great success. We want to thank all of our friends in biotechnology in the Philippines and our hosts here at SEARCA. Our friend Jerome there, thank you for always supporting what we do. And I wish this event all kinds of success today. Thank you for coming. Maraming salamat po.
Maraming salamat, Mr. Hansel. And as mentioned, we're giving away some prizes. So I don't know what this is, but this one looks fancy. And this one looks also fancy. So we're looking forward to your questions later. You can take some photos and post it online. Don't forget to use the hashtag SEARCA8ADSS, right? Okay, at this point, we'd like to invite everyone on site to please stand for a group photo with all our guests here. For the participants joining online, please kindly turn on your camera. We're gonna take a few shots. Okay? All right, thank you very much. For the Zoom participants, we've taken their screenshots already. Okay. We over 100 joining us via Zoom and Facebook. Okay, so before we proceed with the presentations, we're gonna have a quick pulse check. We have a Menti question. Okay, let's please head over to menti.com and enter the code 86211609. You can also use the QR code for your convenience. There's a complimentary Wi-Fi here at SEARCA, so please connect to SEARCA Guest and try to head over to menti.com, use the QR code, or enter the code 86211609. So we have 3 questions there. The first one being, please cite a crop or a livestock that is currently being developed for improved productivity and climate resiliency? So we're waiting for your responses. Cattle, BT eggplant, rice, banana. Oh, these are individual answers, right? Okay. Oh, we're seeing lots of corn, eggplant. Okay, keep it coming. Next. Excellent. Okay, anyone who would like to share his or her answer? And we might give you this special prize. Anyone from the audience, on-site participants? Oh, yan. The guy in black. Sir, please use the microphone. Anong question? Ikaw ang magbigay. Oh, okay.
Hello. Hello, good afternoon, everyone. I am from IPB.
IPB.
My answer is eggplant.
Eggplant, alam na alam. All right, dahil dyan, you have 2 prizes. Both from USDA and from SEARCA ata. Okay. Okay, yes, congratulations, sir. Next question. Please cite a biotech product that is currently in the Philippine market. Are we looking for 1, 2, 3 answers? Okay. Corn. Golden rice, corn. Okay. Golden rice. Okay. No more prize. So thank you for answering. All right, last question. What do we need? Ayan, pang Miss Universe. What do we need to improve policy development or regulation of biotech products? So we're gonna give you a few minutes to ponder your answers. Wow, one word that sums it up. Budget, public consultations, funds, streamlined processes, funding, of course. All right, so any volunteer who would like to share his or her answer, we're giving out a prize to anyone who would like to share his or her answer. Okay, so we have, okay, the guy in white. That's you. You may please use the microphone and please introduce yourself and your affiliation.
Hello. Hello, everyone.
Yeah. Yes, we can hear you.
Yes. My name. Hello, everyone. My name is Chiao Chuan. I'm a graduate student and I'm also a SEARCA scholar. And to improve policy development or regulation of biotech, I have answered the budget. I think the budget is the most important.
All right. The budget, probably the answer of 80% of the participants. Thank you very much for your response, and for that, you have a prize. All right, thank you very much for all your answers and for all your participation. Now we head to the presentations for this afternoon. First, we're going to talk about crops innovations in the pipeline for productivity and climate resiliency. And we have with us one of the leading scientist lecturers on agribiotech in the Philippines and the region, as well as on the science, applications, and regulation of biotechnology. Let's welcome back the Executive Director of ISAAA and the Director of the Global Knowledge Center on Biotechnology, Dr. Rhodora Romero Aldemita.
Yeah. Thank you once again. Okay na ba? Can you hear it well? Okay. Yes, okay. So this is now our presentation, and we'll start with crops. Actually, biotech crops have been developed and 3 decades ago, and since 1992. No. Since 1992, some crops have been commercialized. And so I'd like, in the beginning, yun. Okay. Which is better, yeah. After I didn't wear my contact lens, I forgot. So why biotech? Why are we having biotechnology? First is because of the high global population. It has been increasing. And with the current food production, the status of food production globally, it seems that we have to increase our global food production by 70% in order to feed the increasing population. We also have a problem on climate change, which we are experiencing right now. And this, both the increase in population and climate change, results to severe food insecurity and undernutrition in low-income countries. The current data shows that this was 2019 data, but currently there is over 1 billion already suffering from hunger, and they need to achieve. We need, what is this? Need for biotech. We need biotech so that we can meet the Sustainable Development Goal number 2, which is to have a zero hunger by 2030. And it looks like business as usual attitude in the developing countries, in the developed countries, will not help us achieve this zero hunger. So why do we need biotechnology? Those are the reasons why. And biotechnology is one of those instruments that we can use to meet these problems. And we know that we have 2 kinds of biotechnology: the conventional biotechnology, which uses fermentation and the use of living organisms, the whole living organisms, and the use of modern biotechnology. Now, modern biotechnology uses DNA. Of course, you know this, that they contains the genetic code of life, and that all organisms contain the sequences of nucleotides, and they can be interchanged. Just like what happened to our BT corn, Bacillus thuringiensis corn, where we got the genes from the bacteria, Bacillus thuringiensis, and we put the gene inside the corn, and now we have it also in eggplant. So what we have from 1992, which is the start of the commercialization, but the big commercialization, the global commercialization started in 1996. And we have already products of biotechnology that fight diseases, that increases yield, protect the environment because we're not using any more very hazardous chemicals. And we improve crops already because there are genes that target yield, resistance to insects and pests. And we also have more nutritious food, just like the beta-carotene enriched rice and many others. And we help the industry. This is when we use biomass to produce biofuels or some plant-based products that can be used for the industry. So currently, or since 1996 to 2019, we have recorded this number of hectares, 190.4 million hectares, planting biotech crops all over the world. And may I tell you that ISAAA has been compiling this data from 1996 till 2019. So ISAAA has been the authorized or an authority in producing this data. And by by 2011, we see that developing countries has surpassed the planting area of the industrialized countries. So there are now more developing countries planting biotech crops over the industrial countries. Why is it so? Because our developing countries see the benefits that biotechnology can bring to their countries, to their livelihood, and etc. And I'm going to discuss them with you in a little while. So what are the traits? In the beginning, we had BT. When you answered BT corn today, biotech crop or biotech corn that is planted in the Philippines is not BT alone. It's a stack trait. So when you are asked again, because you will be asked again, I think later, you will just have to answer GM corn or stock trait corn or herbicide tolerant insect resistant corn. And there are still a lot of traits that are already being put into corn together with the other traits. So in the beginning, we just had very low areas planted, but we see that it has increased. But by 2019, the herbicide tolerance trait, which should have been dominating the traits, has already gone down, and this is replaced by the stock traits, meaning herbicide tolerant and insect resistant together. And we see here also the insect resistant ones has plateaued, and there are some pyramiding. So our scientists are still looking for other insect resistant traits, because of course there are pests that attack the roots, the leaves, and of course the fruits, the grains, etc. So our crops should have all those genes so that it can protect themselves from all the different pests. So this is the data, and we are putting now together the 2020, 21, 22 data for publication very soon. Knock on wood. And so, what are the countries which have planted biotech crops? In 2019, this is our data, and you see here that the Philippines is number 12. So we have planted close to 800,000 hectares, and there were 10 Latin American countries, 9 Asia Pacific, 2 North American, 2 EU, which is Portugal and Spain, and 6 African countries. And by next year, when the report comes out, we will see that there are more African countries that have planted biotech crops. So this shows that there are more developing countries. So if you can look at the asterisk, not the asterisk, the italicized ones, or the one with the italics. Yeah, the one with the italics. There are more of those, meaning there are more developing countries planting biotech crops. Sorry. Okay, what am I doing? That, okay. So now let's look at the traits. So insect-resistant traits. We have them deployed in all of these crops, and we have them in the Philippines, maize, cotton, potato, and so on. So we have eggplant there, and cowpea in Africa, sugarcane in Brazil and in Indonesia, and many others. We also have, so there were a total of 307 transgenic events with insect-resistant traits. So these now help us to understand why developing countries, or why countries are planting biotech crops because of these traits. Crops are now resistant to insects. Second are the herbicide-tolerant traits. So this will help our farmers cultivate crops much, much easier than before, because when crops have herbicide-tolerant traits, they can easily get rid of the weeds by just spraying herbicide, but the crops are standing and only the weeds are dead. So there were about 361 events that were deployed in maize, cotton, Argentine canola, soybean, and 10 other crops. And these are the common herbicides, Roundup Ready and glufosinate ammonium. These herbicides were thought or are classified as a class of herbicides that are not hazardous. They are very user-friendly. Once they are sprayed in the field, they can just kill the weeds and are not toxic to any other microflora in the soil. So they easily degrade after a week. So it doesn't pollute our groundwater, or it doesn't affect other crops. And then we have other biotic stresses. Biotic meaning they are living organisms. So biotic are the diseases and pests, other pests. So we have late blight resistance in potato, nematode resistance, virus resistance, very important. You see there are a number of events, is 27. So we have papaya, squash, and potato. We have beans and many others. Okay, now what do we have in the Philippines? In the Philippines, we have these crops approved for cultivation, and we have been cultivating biotech or GM corn, which has the stock trade since 2002. Rice, our golden rice, has been approved for FFP, food, feed, and processing since 2019, and cultivation in 2021. And for eggplant, this has been approved for FFP in 2021 as well, and cultivation last year. So everything is good. We are hoping that our rice and our eggplant will be out in the market. We are also receiving a lot of applications for more traits in our corn, because technology developers do not stop in developing new transgenic events in corn. Now for food, feed, and processing, these are the crops, and these are all, what do you mean by processing? So for example, we have Argentine canola, of course you know canola oil, and maize for our food and also for our oil, for fructose, and many others, and soybean for soybean oil, for soy sauce, and etc. Okay, so we're happy that the Philippines is #1 in Asia in terms of cultivation and in approving biotech crops for food, feed, and processing. Now here is a data which has been published last year by Graham Brooks, and this shows to you what are the contribution of biotech crops. So as I said, they have been planted since 1996, and since that time, this is the recent data, there has been a production of 1 billion tons of food. In 2018, it was only 822 million tons, but now it is 1 billion tons. You can look at it at their website, Graham Brooks, you can easily get it from their website, and this will show you how much productivity is there from biotech crops. In terms of sustainability, that means if you have produced this number or this biotech crops, you don't need much land. You don't need a lot of land to produce this much food. Because for example, this is the data in 2020. In order to produce 85 million tons of food, you will need to have 23.4 million hectares of land. So you see, you save land. You don't really need to cut off the trees, denude your forests, reclaim lands, because just planting biotech crops, you can produce food in such a smaller amount of land that you don't need to get some more, okay? All right, so for climate change mitigation, this is a new data from 1996 to 2020, and there is a reduction of 748.6 million kilograms, a reduction in the use of pesticides, and the global reduction of 7.2% on the area planted to GM crops. So there is a decreased use of crop protection products because our crops already has the resistance. So you don't spray anymore, you don't spray, you don't get exposed to pesticides and all that. And we also have reduction in carbon dioxide emission. By 2020, the data showed that it saved 23.6 billion kilograms carbon dioxide, equivalent to removing 15.6 million cars off the road. So this is very big. You see, you know that cars contribute to global warming because of its carbon dioxide emission. And so with the reduction in the number of cars off the road, which is, 23.6 billion kilograms. So this really affects or give a positive impact to our environment and reduces global warming. So that is climate resiliency. And the same data in 2020, our farmers have obtained so much benefits of $103 per hectare. This is in addition to what they have. And for 1996 to 2020, $261.3 billion have been obtained from biotech crops, which is equivalent to $112 per hectare. It's very big. Even me, I cannot imagine how much benefits the farmers have in terms of extra income and global farmers' income. And this alleviate poverty and hunger by 17 million farmers, including their families. Every year, we collect this data, and we see that with the increasing area planted to biotech crops, we are hopefully getting more farmers and their families to be benefited by using biotech crops. So I have addressed our topic today: food security, sustainability, and climate change mitigation. Now let us see what are the other. Okay, so this is what the Philippines had benefited from. So 2020, about this much. This is only for herbicide-tolerant maize, and for insect-resistant maize, this is much higher than the one recorded before. So that's why our farmers are still planting biotech corn. Okay, so the Philippines, as I said, are planting, biotech corn. And so I have discussed this, and these are now the Pinoy biotech products. We have golden rice, we have EFSP, eggplant fruit and shoot borer resistant eggplant, and BT cotton. So hopefully our farmers will get to enjoy the benefits of using these technologies very soon. So now let's go to, I have discussed with you the GM, genetic modified crops. Let's move on to the new one, the new innovation, which is gene editing. So we have discussed GM, and now gene editing is when there are three types. You can just cut and repair, and then you can cut and use a template, and you can cut and insert a DNA sequence of DNA. So you have there three types: SDN1, SDN2, SDN3. Yeah? Any yes? Because I don't want to expound on the details of the molecular biology on that. But if you want to look at, or if you want to read more, you can go to the ISAAA website. We have gene editing resources there at the website. We also have a book published in 2020, and we are publishing every twice a month a gene editing supplement, where all of the news coming from all over the world are collected and published in a very simple way to lead you to the original article. So many of our academicians, member of the academy, are looking at this resource so that they can, impart the information to you. Okay, so the first gene-edited product was high oleic acid soybean oil. So this has been produced in the US and commercialized in 20, do we have that? Okay. Early in 2019 or earlier than that. And then we have GABA tomato, which now produces substances that will lower down the blood pressure. So this is already being commercialized in Japan. You can just buy it online, and there are also some studies on how much tomatoes are you going to eat so that it will be effective in your therapy to reduce high blood pressure. So this is now something that has revolutionized the acceptance of biotechnology in Japan. The formation of GABA tomato, and of course some of the fishes, which I'm going to tell you in a little bit. We also have, so these are now the ones in the pipeline, and camelina, which has 5% increase in oil. I'm just going to enumerate to you these ones. Canola, which has shorter and more pods, and they become easier to harvest. We have cherry tomato, which is resistant to pepper veinal mottle virus. So we are now looking at disease resistance. And in wheat, we have resistant to powdery mildew. For disease resistance as well, we have banana, resistant to banana Santomonas wilt. In East Africa, banana, as we all know, is a staple in Africa. And canola, resistant to white mold. Here we have tolerance to abiotic stresses. Abiotic stresses comes because of climate change. And here we have drought tolerance in sugarcane, drought tolerance in corn, drought tolerance in soybean, and yeah, many others coming up because they know that we will have more drought in some places. And then we also have lower lignin content so that the leaves of crops can be used for silage. The presence of lignin doesn't make the silage digestible. So once lignin is not there, so it's easily digested by our ruminants. We also have barley with delayed germination because of precocious germination in some crops, because of abiotic stresses as well. And we also have gene-edited crops for consumers. So this is for us, for everybody who wants to eat. So we have sorghum with fragrant, improved fragrance for food, liquor, and vinegar brewing. We have potato with decreased cold-induced sweetening, reduced acrylamide formation, and prevents food wastage. So they're all coming up. And who knows about food wastage? We know that when crop or when potato, when apples, when bananas turn brown, you don't want to eat it anymore, you just throw it away. So this is why we have been focusing on consumer traits so that we can reduce food wastage. Okay, other more consumer traits are the increased sugar content in tomato, the source of natural sweetener inulin in chicory. So this contains, they no longer contain bitter compounds, making the processing cheaper, easier, and more sustainable. Wheat with reduced asparagine and carcinogenic acrylamide during baking and toasting. Leafy greens, so these are new ones from Pairwise. This is a Pairwise company, and they are now developing gene-edited leafy greens with rich green and deep purple colors. So that makes anthocyanin as well in their lettuce and their greens. They also have berries, blackberries, and other berries which are seedless. You can just imagine eating cherries without the pit, or raspberries where it doesn't go to your teeth. You know, it's easier to eat without those small seeds. And so let's look at what has been happening since 1996 to 2021 in terms of the crops that are gene edited. So rice, since it's a staple of more people in the world, I think it's 90 something of the global population eats rice. So that's why the focus is on rice, followed by tomato, maize, soybean, and other crops. So we have gene editing has been applied in 63 different crops. So as soon as the science flourished, our scientists started to work and develop these gene edited crops. with different traits. And the country which has the most research is China, followed by the US, the EU, and so on. So in Asia, we have China, Japan, India, and Australia, who are striving or having a lot of publications, researchers on gene editing. Now, what's happening in Philrice or in the Philippines? We have gene editing, and this is now the reduced browning banana, the first gene-edited crop determined as non-GMO only 3 or 4 months ago. So this is now available, and as soon as the procedure comes, then our farmers can avail of this reduced browning banana. So we also have research, upcoming research at the Institute of Plant Breeding on high lycopene tomato, low phytate corn. At Philrice, of course, and at Erie, we have rice being improved for optimal amylose content, resistance to diseases like bacterial blight and tungro, and improved yield. Now, this is what I'm talking about. So in fish, we have a lot of advances as well. This is now an example in Japan. We also have some in China. So this is now gene-edited red sea bream and gene-edited tiger puffer, which increases their mass. You see, they're bigger and they mature earlier than their conventional counterparts. So this will really help food production and food accessibility globally if it is distributed globally. Now, so the potential benefits in agriculture of our gene-edited products, crops, we have, this is another tool that will improve the pace and scope of innovation to deliver better seed products to our farmers. Dr. Hugh Jones will discuss the regulation. What are the regulatory policies so that everybody, consumers, and everyone in the planet can benefit from gene-edited products, gene-edited crops. So these are now the potential applications in crops. And so in the Philippines, we already have developed the regulations for GM initially, and then now for new breeding innovations, techniques was in May 2020, and it has been approved last year. And now we have the draft animal biotechnology guideline for GM animals. And we are currently under the comment period. And so these are, again, the Philippine institutions with agri-biotech research. You can see yourselves there. You have the Institute of Plant Breeding, Philrice, Erie, PCC, with the Livestock Biotech Center, Bureau of Fisheries and Aquatic Resources, and the Philippine Fiber Development Authority, and some of the research institutions, the UP system, the UP, the researchers under the UP system. So this is my final slide. I'd just like to emphasize that due to biotech, we are expecting this much of productivity in corn. How much more in other crops? I have already defined to you, identified to you what are the crops planted and developed through biotechnology. And in corn, this is already what we have, what we are expecting. So we are hopeful that we can get the SDG 2 by 2030 if we use biotechnology. Okay, thank you so much for your attention.
Okay, thank you very much, Dr. Aldemita, for that comprehensive presentation. So I'm pretty sure if some of you have questions, would like to remind our audience to please write your questions on the papers to be distributed. Oh, it has already been distributed. For our online audience, you can type in your questions through our chat box. So anytime. So while the presenters are giving their keynotes, you may already type in your questions, and my colleagues will be collecting them later. Okay. So from crops innovations, we now move forward to the livestock innovations in the pipeline for productivity and climate resiliency. We have with us this afternoon a strong advocate of biotech as an alternative tool to increase the livestock production of the country towards food security and sustainability. We have the director of the Philippine Agriculture and Fisheries Biotechnology Program. Ladies and gentlemen, I'd like to present to you Dr. Claro Mingala.
Thank you very much, Sunny. The title of my presentation is Livestock Biotechnology Innovations in the Pipeline for Productivity and Climate Resiliency. Good afternoon again to everyone, and I hope I can impart to you some of the information that I gained throughout the time that I've been with the Philippine Carabao Center, of course, the Livestock Biotechnology Center, and now the director of the Agricultural Biotechnology Office of the Department of Agriculture. Okay, so I won't be detailing much of this slide because Dr. Aldemita already mentioned about the very reason why we have the biotechnology, of course, to feed the world. As Mr. Mark mentioned earlier, feeding the world sustainably by 2050 in a global challenge that requires a comprehensive approach. Moreover, there is an urgent need to reduce the environmental impact of livestock agriculture while increasing its productivity. So this particular slide is actually telling us so much that we need to understand why we need biotechnology. But of course, biotechnology is not the only option. As I always emphasize when I present about biotechnology, this is just an option, no? That can be used to increase the productivity of our agricultural products or agricultural resources. So this impact of climate change on food system, I think it's also mentioned to us by Dr. Aldemita earlier. As the climate change is increasing, the risk of natural disasters, including droughts and floods, which in turn is having a massive impact on global food systems, contributing to a rise in global hunger and threatening the livelihood of millions of farmers, herders, and fisher folks. Probably, it's more common to us to see climate change affect crops. No? Not probably seldom we can relate climate change to livestock, but if we are into the livestock industry, they are also prone to climate change, especially if the area where they are being grown is flooded or prone to drought. No much of like source of feed, no? Or forage, and as well as the changing of climates would also bring disease, no? Changes in the disease occurrences, maybe because of the patterns of rains or even the length of summertime, no? During the season of the year, throughout the season of the year. So not only crops, livestock, and fisheries as well. No? We always see in the news about the fish kill, no? That are being delivered to the news nowadays because of probably changes in the temperature of the ocean or even the occurrences of high level of toxins in the seas or even in the lakes. Meanwhile, the growing food security and security situation in the Philippines is also a serious concern that affects millions of people, especially the poor and vulnerable. Probably the rich one can buy their own food or doesn't, give much attention with regards to the sources of their food. So the main reason or the people that are mainly affected are the poor and the vulnerable. Food insecurity means that the people do not have enough food or access to nutritious food to meet their dietary needs and preferences. There are some stories that even salt can just be the, the food they can eat with the rice. So it's really very difficult if you are among the marginalized. It is also not only provides a significant source of protein and essential nutrients for human consumption, it also contributes to the livelihood of millions of people worldwide, particularly in developing countries. That's why livestock production plays a crucial role in achieving food security. And in the Philippines, the livestock industry is a vital component of agricultural sector. There are always discussions in the Senate. Probably you managed to watch in the TV during the deliberations in the Senate with Senator Villar, and the rest of the management of the Department of Agriculture when they're pushing through the advancement of livestock sector in agriculture. So there's a lot of issues and concerns that we need to tackle. It contributes around 18.23% of the gross output value in agriculture and provides livelihood to many people living in rural areas. Currently, the industry faces various challenges due to disruptions in production brought about by the coronavirus, of course. Not only the livestock sector, but the whole country. Alongside transboundary animal diseases, the like of avian flu or the African swine fever, environmental issues and market competition. So those issues and concerns are really very essential factors that affect the livestock industry in the country. So in here, of course, as I mentioned earlier, biotechnology can be an option, no? And also mentioned earlier by Dr. Aldemita already. I won't be detailing this slide anymore. But in agriculture, biotechnology empowers us to manipulate the intricate mechanism of life to optimize crop and livestock production. So biotechnology has contributed immensely to increasing productivity in the livestock industry, particularly in developed countries. It can help achieve food security, reduce the threats of diseases, and ensure environmental sustainability. For example, biotechnology is being harnessed in various aspects of the livestock industry to hasten breed development, to improve animal health and welfare, enhance reproduction, and improve nutritional quality and safety of animal-derived food. Artificial insemination is also part of biotechnology. It allowed significant genetic improvement, productivity, as well as the global dissemination of selected male germplasm. Embryo transfer provides the same opportunities as well for females, albeit on a much smaller scale and at a much greater price. Molecular DNA markers can be used for genetic improvement through marker-assisted selection, as well as to characterize the conserved animal genetic resources. So this is a very famous Dolly the sheep, Dolly the sheep, the cloning of a sheep in 1996, around 1997. One of the most notable breakthroughs of animal biotechnology, the first in mammals, they said, but there are some argument when they said it's actually a sea urchin that is the very first cloned animal, and then the mouse. I don't know if you know, if you remember Cumulin the mouse, which is, they said it's actually the very first animal that was cloned, not Dolly, but Dolly is much famous if we talk about cloning. She was cloned by associates of the Rosley Institute in Scotland using a process called somatic cell nucleotransfer. The successful cloning of Dolly led to widespread advancement within stem cell research, including the discovery of induced pluripotent stem cells. It also dispelled decades of presumption that adult mammals could not be cloned, and igniting a debate concerning the many possible uses and misuses of mammalian cloning technology. So this is the story of Dolly the sheep. This has opened up new possibilities for medical research as well, including the potential for cloning or using cloning to produce organs for transplantation. So I mentioned here medical research because animal biotechnology would not only entail about agriculture or food production, but also production of bioreactors for drug, for therapeutic application, and as well as probably mimicking the disease progression that could lead to the discovery of treatment for a particular disease. So the Philippine Carabao Center has adopted the somatic cell nuclear transfer technology to complement other existing reproductive tools for buffaloes. Probably you heard the news about the Philippine Carabao Center producing the somatic cell nuclear transfer water buffalo. But there's also some difficulty or hardship that we encountered during that process. Buffalo cloned embryos had been successfully produced in vitro. I said in vitro because we were not able to manage to deliver the offspring yet using this technology. It's very difficult. It's not that easy. So you should, if ever you will be able to produce the embryo in vitro, you need to be also very successful in injecting or introducing that embryo to a surrogate mother so that it can become an offspring. But it's difficult because probably more optimization should be realized first before you can get the real offspring of a somatic cell nuclear transfer water buffalo. The present work is conducted to develop and optimize a system for cloning through somatic cell nuclear transfer in water buffalo by the Philippine Carabao Center. But some countries, including India or China, they already claim that they already produce water buffalo through this particular technology. And we don't know how true, but probably true. But this particular technology is also very promising in terms of livestock production. Moving on to the modern technological innovations used for livestock breeding, cell-mediated genome editing refers to the use of cells to introduce precise modification to an organism's genome. This is similar to what we are doing in crops, as mentioned earlier by by Dr. Aldemita. This technology has significant potential as a new breeding innovation for livestock production. So not only genetic modification, but as well as gene editing. By introducing precise modification to the genomes of livestock animals, it is possible to improve traits such as disease resistance, growth rate, and meat quality. For example, researchers have used CRISPR-Cas9 to produce pigs that are resistant to porcine reproductive and respiratory syndrome, or PRRS, a devastating disease that causes significant economic losses in the pork industry. So it's a viral disease, actually. In addition to improving production traits, cell-mediated genome editing can also be used to enhance animal welfare. If I mentioned it right, the PRRS pig was also developed by the one that developed Dolly the pig. The institute, the Roslin Institute in the UK. But they have an equivalent team in the US that also doing gene editing for pigs, particularly in this particular disease, yung porcine reproductive and respiratory syndrome disease. For example, researchers have used this technology to produce also hornless dairy cattle. So this is being done by UC Davis, and eliminating the need for painful dehorning procedures. Unfortunately, the FDA of the US was not able to approve the application of this particular technology, the hornless dairy cattle, because of a portion of the plasmid that was used left inside the animal. If we say gene editing, there should be no foreign genes. So when they were able to detect part of the plasmid that was used for inserting or editing the gene, they disapprove it. And Dr. Alison Eninam is the one that the project leader of this particular project, no, was very disappointed because they cannot actually convince the FDA that this particular technology is gene editing, not genetic modification. But of course, they need to follow the definition of gene-edited animals. There should be no foreign gene that is inserted or left inside the organism. No? So gene editing has been found to be a valuable tool to for making livestock to better adapt them to rapidly changing climatic conditions. The slick hair coat is a genetic trait that gives cattle a short, slick coat, allowing them to better tolerate hot weather. So this is also part of addressing climate change. No? This trait occurs naturally in some cattle raised for food in tropical and subtropical climate. Through the CRISPR-Cas9 gene editing technology, the slick coat trait was successfully introduced into the beef cattle through the alteration of the prolactin receptor gene to shorten the prolactin receptor protein, resulting in a slick coat trait. The slick coat trait is significant because it can help improve the sustainability of beef production and improve animal welfare in warmer climates. Cattle with this trait are potentially able to better withstand hot weather and reducing temperature-related stresses and potentially improving food production. Next, high-producing Holstein Friesian dairy cattle have a characteristic black and white coat, often with large proportion of black. No? Compared to a light coat color, black absorbs more solar radiation, which is contributing factor to heat stress in cattle. This can negatively affect their production levels, fertility, and welfare. Using genome editing, a 3-base pair deletion was introduced in the melanosomal protein 17 gene, which is involved in pigment deposition and polymerization in the pigmented cells. So these are just some of the examples of gene-edited livestock that are now being, let's say, optimized because they are not yet commercially available. They are still probably we can call it laboratory stage or experimental stage, but they already able to produce the animal. No? The edited genotype now represents the valuable model for studying the impacts on heat tolerance and determining any potential effects it might have on performance, milk production, and resilience to disease associated with exposure to sunlight. So gene-edited animals in the pipeline. Additionally, through genome editing, it was made possible to generate hornless cattle. I already mentioned this earlier. And then we have the gall-shaped pigs, which are a line of domestic pigs that have undergone an intentional genomic alteration to eliminate alpha-gal sugar on the surface of the pig's cell. Ano ba yung alpha-gal's, gall-shaped, alpha-gal sugar? Then we call it gall-shaped pigs because this is a significant because people with alpha-gal syndrome may have a mild to severe allergic reactions to alpha-gal sugar found in red meat. So it's really very important, especially those people that are meat eaters. No, the FDA has approved gall-shaped pigs for both human food consumption and as a source for potential therapeutic uses. So they're targeting two objectives, no, for food consumption and the source of potential therapeutic uses. Gene-edited animals in the pipeline also includes the Red Angus females, which have an excellent milk production and have a strong maternal instinct. This breed produces a highly desired carcass with the meat being of excellent quality. This is due to the intramuscular marbling. The significance of this development is that it expected to create new genetic opportunities and markets for red Angus cattle. This particular product is also another example of gene-edited animal that would be the black to red Angus animals carrying gene-edited traits for heat tolerance and coat color. So as I mentioned earlier, if it's black, it absorbs more sunlight or ultraviolet that could lead to stress. So that's why they are now using it or towards the color red. So, but also similar production traits as before, no? Then we have the gene editing has been used to create pigs that are resistant to PRRS, as I mentioned earlier, no? Because the virus attacks young pigs and estimated to cost farmers around 2.3 billion a year. I think a team from France also using gene editing now to address the African swine fever. So they are trying to develop a resistant pig line from African swine fever through gene editing. But this is a very now it's in the preliminary stage. But you can probably Google it. Probably it's already in the web how they are developing that pig line. No? So this is a significant development as it could greatly benefit the food and agriculture industry by improving animal health and reducing costs for farmers. So aside from pigs, there is also poultry that is resistant to bird flu. All of these are still in its preliminary stage. Probably using biotechnology in livestock is not that much easier than crops, probably because of they said it's more complicated. I don't know actually how they are doing that in crops, because probably they can raise the crops in a very short period of time and in a confined and controlled environment, easily done compared to livestock. Okay, so this one, of course, while we are promoting or pushing for the use of biotechnology as an option for agricultural production, at the Department of Agriculture, we are also putting the regulatory policy, of course, so that the risk can be minimized or even not only minimized, but to really avoid, no? Especially with regards to the environment, animal welfare, and the public health. So that is why the GM animal regulatory policy are being reviewed now, the draft, and we are looking forward that this will be signed very soon by the 5 departments: the DOST, DA, DENR, DOH, and DILG. So all of these departments has their representation to the technical working group in drafting this particular policy. No? That's why we call it the Joint Department Circular. So ways forwards to ensure enabling environment for biotechnology undertakings, implementation of a clear, predictable, science-based and risk proportionate regulations. Probably this will be elaborated more by our speaker later on. Establishment of an adaptive and responsive policies that can adapt to rapid advancements and emerging technologies. Foster international cooperation and harmonization of regulatory standards. Streamline global biotech development and facilitate cross-border research and trade. Adequate sufficient funding for biotech research and innovation, and education and public awareness. I think that's all. Thank you very much for listening.
All right, thank you very much, Sir Bong. We've already received some interesting questions from the audience, so we hope that later we're going to have some more time to engage with the participants. Okay, so right now we're at over 200 online participants and still over almost 40 participants on site. And online, we have our resource speaker joining us from the Translational Genomics for Plant Breeding of Aberystwyth University. Do we have Dr. Yu Jones with us now? Okay.
Hello, good afternoon. Can you hear me okay?
Yes, sir. Loud and clear. All right, sir, you now have the floor.
Okay, I'm sharing my screen. Are you seeing now my full slides?
Yes, sir.
Excellent. So good morning. Oh, well, it's good morning from me. It's good afternoon to all of you in the room. My name is Hugh Jones. I'm speaking to you from the beautiful West Wales coast of Aberystwyth. I'm a professor of essentially plant molecular genetics here at Aberystwyth University. And I'm going to be talking to you about targeted plant breeding done through new genomic techniques like genome editing and the tension between innovation and regulation. So just a quick slide showing some of the work that I do. I use biotechnology techniques to understand expression, gene expression and plant development, so how plants work at a molecular level. But I also, of course, understand the strong potential of biotechnology in plant breeding. And apart from my role as a scientist at a university, I work for two government agencies in the UK, the Food Standards Agency and the Department of the Environment, in various expert committees that develop policy and risk assessment for these kind of products. But I'm not speaking for those organizations. I'm speaking as a research scientist here in this meeting. So like the speakers before me, I'm just going to quickly outline some of the global challenges of agriculture, and I've divided them into 4 different categories, but you can cut and splice these challenges in lots of ways. So agriculture, global agriculture, has to make more nutritious food using either the same or less land. We certainly don't want to put more land to agriculture in the future. And we have to do that under an ever worsening scenario of heat, cold, pest, disease pressures. We'd like to do that. In fact, there is a big call from consumers to do that with less inputs of fertilizer, pesticides and water and so on. And of course, agriculture has to play its part in lowering the greenhouse gas emissions that agriculture and the food system currently is responsible for. So how can we do this? Well, it's my belief that plant breeding can play a part. So the more advantageous genetics we can combine into a seed before it's sown into the ground will make that plant more able to meet those challenges we saw in the last slide. But plant breeding is slow, and the techniques of molecular genetics now give us a route that we can speed that process up. We can make it more precise. The changes that we want to make in the plant, we can make them in a more directed way than some of the older methods of plant breeding. However, all of this will only be possible if the regulatory frameworks that control the commercialization of biotechnology are fair and, of course, are safe, but also allow a route to commercialization. Now, the problem with new biotechnologies, the problems they face, is that some of the laws and the basic understanding of biotechnology was set up in the 1990s and haven't really changed since, even though the science has moved forward quite markedly. And I've just shown a couple of quotes of what you know, the core meaning of what is a GMO there on the screen, with various exemptions and details that different countries have added to the basic definitions. And of course, the science now means that we can do techniques called gene editing, and the actual tools that we can use vary. So we have zinc finger nucleases, we have talens, we have the CRISPR-Cas systems, and others. And these are capable of very precisely making changes to a host genome. And that can be an animal, a plant, a bacterium, etc. So even though the tools are all quite, you know, there are a collection of different specific tools, they all have two main things in common. They have a part of the chemical that is doing the editing that can scan the host genome. So let's call those the eyes of the molecule. They can be the target gene can be set by that section of the molecule. The eyes can scan the genome, the host genome, to find that portion and stop as it reaches it. And a second part is an enzyme, an endonuclease enzyme, that can cut the DNA at that point. And all of those different tools, broadly speaking, can do that. Scan the genome and then cut the genome at a set point. Now, those techniques can be used to make a range of outcomes. We've already heard of this SDN type 1, 2, and 3 type of edits. So I've put those onto a kind of a scale that on the left is being defined as more natural, less intervention, that it's harder to detect those changes that are SDN type 1. So these would be, as an example, SNPs, so small changes in single or short ranges of DNA caused by a cell repair system called non-homologous end joining repair, where after the cut is made, the nuclear repair system aims to correct that cut, and often that results in changes in the exact sequence. So at the other end of that scale, we've got the SDN type 3 changes, where not only is a single or 2 or more cuts made in the host genome, then a repair template, like a sticking plaster piece of DNA, is also added into the nucleus at the same time as the gene editor that's making those cuts. And the cell now has a template in order to make a repair, and that template can flip in a new piece of sequence. And under the SDN3 type, that is a functional gene being flipped in through homologous recombination using that template. Now, SDN2 is in the center of that spectrum, and it's still using a template repair, but the template is much shorter, and often it's just a single or small piece of DNA. However, it's more complicated than that, because, of course, we can now use gene editing tools to make changes to the epigenome or to the way gene expression is controlled, and I've called these SDN type 0 changes, where there is no change in the DNA sequence at all. We've also got a slight complication in the SDN3 types, because classically an SDN3 type of edit would include an insert, a functional gene that is not part of the gene pool of that organism. And in most regulatory authorities, that would be defined as a GMO. It's a foreign piece of DNA. It's foreign to the gene pool of that organism, and it's been inserted, okay, into a safe harbor, into a known location, but it's still a GMO. But however, if we use the SDN3 type of insertion to insert an allele of a gene that's already there, then that isn't a GMO. It's what we do using classical plant breeding. So that would be somewhere towards the left. So I'm just trying to emphasize that this SDN1, 2, and 3 type of edit is actually more complicated than it appears. So how have countries taken on board these new tools and adapted their regulation? Well, I've separated those countries into animals, the hares, the tortoises, and the ostriches. So those animals that classically had a long history of cultivating GMOs were the first to adapt their laws to accommodate genome editing. And then other countries, the EU, the UK, and so on, the Philippines, I'm putting in that category, also changed theirs. But more concerningly, there are countries that haven't yet got any new laws to deal with genome editing, and that needs to be corrected, because obviously food is a global commodity in many ways. So this map really shows that. So those countries in dark green have laws that are supporting these new technologies. Those countries in white still have no laws in place. But even though there are laws, we can see that the EU in red there, New Zealand, and to a certain extent South Africa, they have laws, but they are not easy for gene editing to be used. They've largely put even simple gene editing into the camp of a GMO. So the SDN1 and the SDN3 as a non-GMO in the former case, and as almost always a GMO in the second case, largely are those different countries have got, you know, their own laws. They're all worded slightly differently, but they concur on the simple types of edits that are not GMOs and the more complex types of edits that are perhaps targeted GMOs. However, there's a whole area in the center that those countries, there's uncertainty. And I've just got a couple of examples here. So this is the case from Australia, where even so short template repairs, if a template was used, regardless of how short that template may be, is still comes under the GMO law. So only the non-homologous end joining repair type edits would fall clearly under outside the GMO law in the Australian system. The same in Japan, although it's not quite so clear, but in this case here, this interpretation of the Japanese law puts SDN2, so that central category, into the LMO, the LMO laws, so it's not excluded. This is an example from Thailand, where SDN1s have a certain amount of responsibility, but not a full GMO type, and SDN2s have some of the expectations for risk assessment, but perhaps not all, as a case-by-case system. However, it's not clear as an applicant what type of SDN2 change would kick into place the full GMO law and which would be excluded. The EU has just announced a proposal to change its legislation to accommodate gene editing, but in its science detail, we've got these really quite complicated descriptions where only if a substitution or insertion is less than 20 nucleotides would it be excluded from the GMO law. I'm not sure there's any scientific understanding of why this number of nucleotides was chosen. So what are we doing in the UK? Well, the UK has largely sidestepped this issue of SDN 1, 2, 3 and so on by framing those gene-edited organisms that could be found spontaneously or through classical breeding systems as non-GMOs. So the first thing the UK did was to hold a public consultation that closed in spring 2021. They then used a statutory instrument, so this was not needed to have an act of parliament, to exclude field experiments, so experiments that were done by either companies or research institutes, but where the plant was not entered into the food or feed chain. So those type of field experiments where the gene-edited plant could be made through, or the changes in that plant could be made through conventional breeding systems, and they were excluded from the biotech risk assessment system for those field trials. Then in early 2022, an act of parliament started its route through the two houses, and that came out in just March this year, where it achieved royal assent. So that is now a law in the UK. And broadly speaking, what that does is define those gene-edited organisms where the changes made could have been achieved through conventional breeding. So regardless of the SDN 1, 2, or 3, if there's cisgenesis, intragenesis, and so on, if the applicant could convince a panel of experts that the changes made could have been done in theory, they don't have to show that it could have been done in practice, actually make a plant, but if the arguments could be made that that kind of change could have been found either spontaneously in nature if you looked hard enough, or through a classical plant breeding system, then that organism, plant or animal, would be defined as a precision bred organism, a PBO, and that then was excluded from the existing GMO laws. The Act of Parliament also made no plans for labeling of these organisms, but would form a database that was publicly available, where the new varieties of plant or strains of animals would be named on that list if they were developed by this type of gene editing. And the UK system has made it clear that we'd start the regulatory system with animals, and once that was underway, then we'd start the system with plants, and then once that was underway, PBO animals would be also introduced. I'm not going to go through this slide in any detail, but this is the flow chart of how the new system works. So an applicant who has made a PBO plant or animal would approach this expert committee, the advisory committee on releases to the environment, and it's this expert panel that would make a judgment as to whether that new organism is a PBO, a TBO, a traditionally bred organism, or indeed a GMO, and then different routes through the regulatory system from there. So how would this committee define if this organism is a PBO or a GMO, well, it's done by this expert committee, and they've got to be satisfied that there are two things. So any added DNA that would encode the editor, so the TALEN or the CRISPR-Cas has to be removed. So there's no foreign DNA left in the organism, and the changes made could have, in theory, be found spontaneously or using classical methods. And a PhD student of mine wrote this paper in order to kind of support this activity. So it's a paper that defines all the baseline changes that you can find in spontaneously or in breeding, and this will kind of inform the choice here about, is this organism a PBO, a TBO, or a GMO? So what's next for the UK? Well, ironically, here in Wales and in Scotland, we have our own laws on agriculture. So all of these changes apply to England only at the moment. Wales and Scotland are yet to make these changes. As I said earlier, we are taking a step-by-step approach, and we'll first implement the Act for plants and then for animals once that's underway. There still needs to be additional laws put into place, which we expect to be done next year, that will actually finalize all the legal instruments needed. And as I said, the implementation for animals is going to be done later, once some system of animal welfare is taken into account. So what's next for global agriculture and trade? Well, there will be serious challenges for how the organic growers and the certification organizations react, how there will be integration of this in different countries. The serious lack of harmonized global laws for gene editing and labeling will inevitably make friction for trade. We can easily see examples where in one country a particular type of edit would be defined as a non-GMO and excluded from the GMO laws, but in other countries it would be captured by the GMO laws. So this is going to cause issues for trade. The lists of edited varieties would be very quickly be compromised, because as other breeders incorporate edits from other companies' varieties into their own, of course, once that edit has gone through the regulatory system, it's out there. If it's moved from one type of, let's say, I don't know, corn variety into another, it's not going to be needed to go through the regulatory system again. So who's going to keep track of which new varieties contain those edits? That list that governments say they're going to keep is going to be more of a challenge, I think, than they think. And anyway, the fact that we have real challenges in testing or identifying these edits in different backgrounds could make all this irrelevant anyway, because of course you can't regulate what you can't find. Okay, I'll end there. I'm happy to take questions now or in the panel later.
All right, thank you very much, Dr. Jones, for your presentation. A round of applause. Thank you, sir, for giving us the global landscapes on, of course, on regulatory frameworks on breeding innovations, especially for citing a case in the UK. Please stay with us, Dr. Jones. We're going to have the open forum. At this point, we'd like to invite the first two speakers, Mamola and Sir Bong, to please join us in front. All right, and please sit down. to the magical chairs in front. We're gonna wait for Ma'am Ola, but for our online audience, we still have time for you to type in your questions. We're gonna have an open forum, as well as for our on-site audience. We've distributed some papers where you can write your questions, give them to our colleagues. We're gonna try to read them later. All right, so there's Ma'am Ola. And we also have joining us online, Dr. Jones. At this point, we still have around 200 online participants, both via Zoom and Facebook. And now I'd like to call on my colleague from the Research and Thought Leadership Department, the project coordinator of the AFNR Knowledge Platform, to help us facilitate the open forum, Mr. Jerome Baradas.
Thank you, Sunny. Good afternoon, everyone. Good morning, Dr. Jones, joining us from the United Kingdom. If you notice, people have been handing out pink paper. I hope it was 50 peso bills, but it's not. So again, I'd encourage you to write your questions. Don't forget your names so that we'll be able to properly acknowledge you later on, and so that we know who will be the lucky recipients of the prizes. So if you want to ask your question yourselves, we have microphones. Just raise your hand and wait to be acknowledged. Again, for those who are, I don't know which camera is functioning. For those who are joining us online, we have not forgotten about you. We look forward to receiving questions from you. I actually have a live file, and we have them here. I now have 3 questions. Keep the questions coming. Let's make this an engaging discussion. I'd start with someone who is joining from us from Facebook Live. This is addressed to Dr. Aldemita. Dear Dr. Aldemita, no. Doc Aldemita, you mentioned that there is a need to entice students to go with agri-biotech because the more senior generation is already retiring. Aside from forum like this one, what other programs do you think we should do to encourage the youth to be involved in agri-biotech? This one is coming from Jess from the Next Genre Youth Specialist Group of the International Rubber Research and Development Board. Hashtag SEARCA ADSS. Mamala?
What other
programs to entice the youth?
Okay. Well, in the Philippines, we have all the instruments to help us in inviting students to go to agri-biotechnology. And first, we already have the curriculum in place in a lot of the University of the Philippines system. So we have started this in UP Mindanao, we have in UP Manila, UP LB, and there are still others who would want to improve their curriculum to include agri-biotechnology as a major in agriculture. And then the second one is there are several like-minded agencies in the Philippines who are working hand in hand, and we are focusing on activities that would help the Philippines in inviting students. And at ISAAA, we have a lot of materials that explains the benefits of biotechnology, what are the promises of biotechnology in terms of career. We also have, actually, one of my colleagues, this is in Malaysia, they have established this seminar where they go to universities and invite students to go for agri-biotech, and we hope we can do the same. We also have the Department of Agriculture, Department of Science and Technology, which have programs in disseminating biotechnology information for students. Back to ISAAA, we have this Double Helix. This is a magazine for students, secondary schools, and we explain what's happening in biotechnology. We feature scientists, we feature regulators, the technology themselves, and we are coming up with a new magazine, Pinoy Biotech, to disseminate or expose what's happening in biotechnology in the Philippines. So there are still a lot. And if you have needs in your area where you think we need to focus certain students or certain places in the Philippines where we need more information on biotechnology, then let us know, and we will help you, or we can bring this up to our partners so that we can allot or give scientists or provide information sources, resources, and resource speakers in your area. Yun, thank you.
Thank you, Ma'am Ola. There is a follow-up, a very related question coming from our online audience. This one is coming from Grains, one of the student orgs here. Dr. Jones, the question is, what do you think can be done in the curriculum, in the curricula for it to be improved now when it comes, in relation to what you discussed earlier? What are the vital components that we have to work on in relation to training the next generation of scientists, of those who are working on agricultural biotechnology?
Well, thank you. So I'm also a teacher. I lecture in plant, well, a range of subjects, biochemistry, biotechnology at university. But I think the concept of plant breeding, and especially the new types of plant breeding, are really something that younger children need to understand. It is a fact, and it's a sad fact, that many children don't understand the concept of wild plants versus cultivated plants versus those forms of plants that we eat as food today are really not wild. They are the result of thousands or tens of thousands of years of selection, and in the last few hundred years of active plant breeding. So I'd like to see more education and understanding starting much younger in the school curriculum.
Dr. Mingala would like to add to that. Thank you, Dr. Jones.
With regards to the effort of the Department of Agriculture in promoting the agricultural biotechnology, actually the Biotech Program Office has a scholarship for undergrads. I don't know if some of you were able to avail to that scholarship. We have, I think, giving scholarship for agribiotech to UP Los Baños, Visayas State University, University of Southern Mindanao, I think previously CLSU. And the good thing now, just recently, it was signed by the Senior Undersecretary, our fellowship for agriculture. So those young people, or what do you call this, early career scientists, masters and PhD, can avail the fellowship under the Department of Agriculture. It's actually patterned to the DOST scholarship or fellowship, which could like ranges from SG 24 to 28, something like that. So probably you have an idea how much they will they earn from this particular fellowship. So it's just that it's just recently signed, but it should be like published for next 15 days before it will be finally, adopted. So this scholarship is actually long been waited, and now I'm announcing this especially for our early career scientists to avail this particular program. And we also encouraging our agribiotech to engage to livestock biotechnology, because I know there's a lot of people now engaging to crops. Probably it's now time for us to go to livestock or to animal biotech, and as well as fisheries. Thank you.
Thank you, Dr. Clara. Just to add to the scholarship talk, no, over the past 5 decades, SEARCA has granted 1,753 full MS and PhD scholarships and 100 PhD research scholarships to nationals of SIMIO member countries. So that includes the Philippines. I don't know if we have SEARCA alumni here joining us, but we have SEARCA scholars. Hello. Yan. So hindi, what I'm saying is not fake news. We have proof, living proof, alive and kicking. And just the past year, the past school year, we have 14 scholars working specifically on agribiotech. I don't know this coming school year how many we have. So it has always been an interest for SEARCA to involve the youth. No? My colleague Sunny can attest to this interest when it comes to engaging the youth in agriculture. But in relation to what Dr. Claro said earlier, a plug to work on livestock biotechnology. Our friend here, Eljan. Eljan, say hello. Hi. Oh, you know Eljan, Atisel? So he has an observation that there are really a lot of work being done on crop biotechnology and not much on livestock biotechnology. What do you think can be the reason behind this, and how can we encourage them to go to animals and not just crops?
Actually, this scenario is not just in the Philippines, probably worldwide. There's still a lot of things that we need to do in terms of livestock biotech, also in fisheries, compared to crops. You know, excuse me, we have already big companies in crops like the former Monsanto and now Bayer, and then the Danforth, which is a non-profit organization for crops, biotech. But in livestock, I think very seldom, or not even one. I don't know, probably the ILRI is doing kind of research on livestock, the International Livestock Research Institute in Nairobi, but I don't know how big. But most of the researchers that are being done in livestock is, I think, in the academe or universities, like in Roslin in the UK, or even Peerbright. But you know, that's why I'm also promoting to please engage to livestock biotech, so that the Philippines can also start doing our own biotech innovations in livestock.
Just a quick follow-up. Yeah, it was mentioned earlier that the regulation for livestock biotechnology is currently under stakeholder consultation. So this is the first in the Philippines when it comes to animal biotechnology, right? So are there countries that we know of that have been ahead of us when it comes to regulating livestock biotech? You're familiar?
In our when he presented the different livestock in the pipeline, biotech livestock in the pipeline, you can easily discern which countries. So we have Argentina, Brazil, the US. Now we have Japan. But Japan, of course, the gene-edited one is not under the GM regulation, but we see that fish biotech is gaining grounds in Japan, actually. So the other countries, we have, in Kenya, there is an upcoming one, a regulation on GM. And I think those are the only ones, because the Latin American countries, Argentina, Brazil, and there's another one that has GM animal regulation have been placed. They have been in place. That's why the slick cattle has been developed in the UK as well, but it's not, because there is no commercialized biotech animals as yet. So if there is an ongoing policies on biotech animals, then they could have commercialized it right there. But currently, it's only the ones commercialized is only Aqua Bounty salmon, which is a GM in the US and Canada. And then after that, we were hoping that the hornless cow would be next, but then something happened. And then the next one would be the ones in the pipeline, the porcine, the gall safe pigs, and all that. And they're all still US, Brazil, Argentina.
Okay.
Yes.
If I may add, probably some of the countries like what you call that, joined the crops and livestock together in one regulations. But for us, we separated it because the first one that we drafted, the JDC one, is leaning more on crops. So instead of like amending or creating on top of that, we drafted a separate guideline for GM animals.
Dr. Jones, do you have any addition to that?
No, I echo all of those thoughts that have been made. So in the UK, our primary laws will cover both animals and plants, but the various secondary legislation, the detail that we need to put these into practice, we're actively working on the plant side first, and I guess a year or so after that's in place, we will follow that with animal laws. But I absolutely agree, some countries have gone in two completely different directions for animals and plants. Others have combined their laws together and then will adapt the animal and plants under a single act.
All right, thank you. We have a question here from Davao del Norte. We're really borderless this time. This is perhaps a question we encounter most of the time. With the advent of biotech, GMOs in particular, is there a tendency that endemic species may be extinct in the future? Do we have safeguards about this?
This is a question where we have the origin of species, I mean, the place. For example, Mexico, which is the place where corn was originated, they have this ban against GM corn. So it's an act, I mean, the ban is an act to protect the endogenous corn. But actually, you know that in risk assessment, we know that there are risk management, and they can be managed. I mean, the risks can be managed through planting at at different times of the year, or through isolated distance and so on. So even if corn is endogenous in Mexico, they can plant GM corn. So in some of the countries, they have this tendency, but we try to tell them that the risk that they are thinking about can be managed through scientific ways, and hopefully they can understand the scientific basis of all those recommendations. Yeah.
Actually, it's one of the questions here. It's about gene-edited crops and people's health. I remember Dr. Jones in one of his slides about detection. He mentioned that it would be difficult to detect if it's a gene-edited product already. Is that right? So we have science. How about in the United Kingdom? Are there ways that we are trying to ensure that even though these are, ensure the public, the consumers, that even though these are gene-edited crops, they are safe, these are, the risks are managed very well through scientific, through science. What are your thoughts about that, Dr. Jones?
Yeah, so a gene-edited plant or animal can only escape the laws we currently have for GMOs that are really quite onerous. The risk assessment is really off the scale, serious. But if it's a simple type of gene-edited organism that you could have made through conventional breeding, then that would not be covered by the GMO law. So those events, those kind of edits that could be made through conventional breeding could be made through conventional breeding. So they pose no more risk to human health than all the breeding, all the plants that we consume every day anyway, which have been through a plant breeding process. Now, having said that, the UK system is allowing for a risk assessment. So after a genome-edited plant is considered a PBO, i.e. a gene-edited organism that is not a GMO, it'll still pass to the UK Food Standards Agency for a risk assessment. And that's going to be 2 tiers. So the first tier is, this really is something that we don't need to look at. It's something that's absolutely equivalent to any other new plant variety that we have on our recommended lists every day. 3,000 new plant varieties are added to our recommended seed list in the EU every year. So if it's a tier 1 type of edit, it'll just be considered as a new plant variety. If it's got an intentional change in a nutrient or perhaps an allergen or something that the FSA thinks needs to be looked at, it'll be elevated into tier 2. But I have no concerns at all that these types of PBOs are going to pose any more risk to human health, and the key is, than conventional plant breeding. And conventional plant breeding has a history of being safe.
So in the online questions, I'm trying to balance what we receive online and on site here. There are questions relating to information dissemination, like equipping scientists to be able to face the public and explain it to them. Anyone from the team from our panel of experts here can answer this question. What are the things that we are doing so that we'll be able to empower our scientists and to face the public, explain the science, and at the same time dispel misinformation, especially in the age of fake news? Ma'am Ola, would you like to start?
Yeah, with the support of the Department of Agriculture, the DA Program Office, ISAAA has several or a number of activities that help not only the scientists, but also the students and the media in disseminating correct information, science-based information and biotechnology, and the how. How do we impart the knowledge to our audience? So we have an upcoming one in, kailan nga yun? We have a we just have science communication program, which is a project for the scientists. It's an upcoming one. We just had a social media for students. A number of students from Region 1, 2, 3, 4 attended it face-to-face, and they learned the how. How do we do social media? How do we use social media in communicating biotech? Yes, and I said a while ago, I have also mentioned some of the publications that we have published. So I'll just invite everyone to go to the ISAAA website. There are several, there is a number of products that we are sharing to you. We also have cartoons. We also have materials for young kids and packet case. And so ISAAA's activities are really for knowledge sharing to all the stakeholders of biotechnology, not only in the Philippines, but globally. We also have translations. So we have been doing this for the last 20 years, and we have been harnessing the current information platform. So we have started seminars, symposiums, publications. Now we are into webinars, Facebook, social media, and so on. So we are adapting, ISAAA is adapting, depending on the current
And the
information
audience.
communication platforms.
Yeah, thank you. I think Ma'am Ola has already spoken for all of us doing these things on USDA, SEARCA, DABPO. Quickly from Dr. Jones, are there similar initiatives in the UK about this? Or is fake news really as prevalent in there, or not much when it comes to, especially about GE products?
So there has been a number of public consultations and focus groups, especially run by the Food Standards Agency in the UK, these are really to understand the baseline level of public understanding and attitudes. They weren't designed particularly to change attitudes. But I think in the UK, compared to the late 1990s, where we had an extreme reaction to biotechnology, there was a lot of negative media and understanding. I think that has changed a lot. We seem to have turned a corner, and the science. The messages that are coming out of science are now much more prevalent in the media. And I think we are much, out of the EU countries, we are one of the more accepting of biotechnology.
Thank you, Dr. Jones. I'll take a break from my pink paper and ask, yes, SEARCA alumni going to ask your question. Please use the microphone. Identify yourself first.
Hello. Hello. Hello.
Yes, we can hear you, sir.
Good afternoon po. I have a question to Dr. Jones. Okay, so good afternoon, sir. I'm Jedya Bron from the Institute of Animal Science here in UPLB. I have 3 questions po. Okay, first is, by the way, my questions are quite technical. The first question is, why do we call CRISPR and other gene editing technologies as plant breeding innovations? In my field, in my class, I always say na CRISPR is a revolution in biotechnology. It even rivals PCR. So it's a very big revolution. It's a very big biotech revolution. Why is it that it's called plant breeding? Is it more appropriate to be called biotech innovation? Okay, that's the first question. The second question is the regulation of CRISPR edited products. In my country, if it's CRISPR edited and there is no rearrangement, in other words, it is not transgenic, it doesn't add add a transgene, it's just edited. In my understanding, it is not regulated. In other words, if it's edited and it looks like a natural mutation because it's just edited, it's not regulated. And with that, if we don't regulate this, we assume that it will be always safe. So if it looks like a natural mutation, but it is CRISPR edited, is it safe to assume that it will be always safe so that we will not regulate this? Okay, again, CRISPR editing is a revolution in molecular biology, in biotechnology, because of its accuracy and efficiency. So, in my opinion, natural mutations and CRISPR editing mutations, they may look similar, but they have fundamental differences. Number one, natural mutations are very rare. Okay, if you remember, natural mutation is very low. If we can remember Ma'am DR's book, ano bang sabi niya doon? 10 to the negative 6. It's very rare. But in CRISPR, it is very high. Next is natural mutations are random. It is produced by Mother Nature. In CRISPR, it is produced by humans, by us, and we have motives for that. So in assuming that it will be always safe, so that in CRISPR-edited products, we will not regulate, I think, in my personal opinion, it is not a very, it is a risky assumption. It's not a very safe assumption, but it is quite a risky assumption. And my third question is about gene drives. We all know that the very successful gene drives now are CRISPR-enabled. Okay, so I would ask also Dr. Jones, what do you think? How will we regulate these gene drives? Okay, thank you po.
Thank you, sir. So those are 3 questions, but you'll only get 1 gift. Dr. Jones.
So thank you for that rather in-depth set of questions. The first question, I absolutely agree, yes. I apologize if I personally called CRISPR a plant-only technology. Of course, it can be applied to bacteria and animals as well, and it's a fundamental new step in science. So absolutely, it shouldn't be associated only to plants. So thank you for reminding us of that. I don't accept some of the arguments in your second question. I think in any one meiosis, you might say that a chance mutation is rare, but in a field of plants or let's say a population of animals, then actually mutations are extremely common. There will be tens, if not hundreds of thousands of spontaneous mutations in the cells in a field of corn or wheat or whatever the crop. And regardless of how that mutation was made, so if the cause of the mutation was sunlight or other kind of mutagen or a gene-edited cut, then the change, the resulting change, the SNP or the rearrangement, in my view as a scientist, is indistinguishable. The cell doesn't care what made the cut. It's the correction of that cut through either non-homologous systems or homologous systems, but that is the consequence, and that's what is either safe or unsafe, not the cause of the cut that made the mutation. And in my view, if we judge that change, that edit is something that we accept, the risk of that is something that we accept in the food chain anyway, that we have been doing for the last few hundred years since we've been using the current systems for plant and animal breeding, then I don't see that the cause of the cut needs to be looked at in any more detail. I accept your third question that gene drives is a completely different way of using gene editing tools. These are not commonly found in nature, and so they would need a risk assessment that was of a level that looked at the hazards and the risks. But that's not something that we would see as a PBO in the UK at the moment.
Thank you, Dr. Jones. It's now 4:30 in the Philippines. Please allow me to read through last two questions. These are very good questions we are receiving both from online and on-site. We have a good set of participants this afternoon. Just a quick question. I think these are related. Gene editing policy on this approving projects that has a plasmid residue on the carabao or cattle. Do you think that this should change, or is this level of safety just right? This came from the audience here. Would you like to clarify that question? Yes, ma'am. Please. Please approach the microphone.
Hello po. That's my question po, and I'm sorry if I didn't write the name. I didn't know that it.
It's
So
okay. No, tell us your name if you want.
I am Miriam Tagyab, a third-year BS AVT student with the organization UP Labs.
Okay.
So, my question is that some projects like that of our carabao and cattle are being disapproved, as previously mentioned, because there are remnants of the plasmid vector that is used. And the scientist was very disappointed, as you have said. But the policy is that it will be disapproved because there are plasmid vector remnants. But should this policy change, or is it just right that there is that level of safety that we have placed for our projects?
Yeah, okay. Thank you very much for the question. Actually, that's also some of the arguments that are being tackled. Not, of course, UC Davis, because they are the one who developed this particular product. But the thing is, there is a definition, no, that they need to follow. And since this particular product is outside of the definition, so it cannot really pass through that. Second is, there are things that we need to consider for risk assessment. Of course, we need to consider the allergenicity or toxicity of the product, and then the environment effect, and then the animal welfare. If there are some issues concerning these three, so they need to address. For example, if particular insert would create a protein that would cause allergenicity or toxicity, then they need to assess that or evaluate that, because it's probably create another protein because of that insert. And then, of course, I don't know if it would cause invasiveness in the environment later on that would transfer to the cattle or carabao in the area. So, but of course, animal welfare-wise, natanggalan siya ng sungay, and probably it addressed, the mechanical dehorning process. So probably benefit to the animals, but the two other things they should evaluate. Thank you.
All right, thank you, Dr. Bong. Last one from the College of Economics and Management. I'm always thrilled to see people joining our sessions that are not from Agri. No? It proves that no matter where we are from, I'm from the College of DevCom, hello. And now we have from SEM, it's a question about gene-edited organisms. What if the trait, a good trait, exceeds the regulation? Do we adjust the regulation? Am able to process the question? This is from Rimpol Monreal. Where are you? Would like to ask the question yourself? So I was able to catch your question properly there. What is it possible? Have we encountered this before, that from the field, the experiment, it exceeded in a good way the regulations, and what do we do with this?
In regulation in the Philippines or globally, we always look at what they call as future proofing, that whatever the regulation is always reviewed. And so if there are new innovations, then the regulation can adjust. Different from the other countries where they have laws. Laws are very difficult to change. So in the Philippines, we have EOs, we have ano yun? Memorandum, yeah, yes. And so it's circulars. So for whatever we see, that's why we always monitor the Bureau of Plant Industry and the Bureau of Animal Industry in the future will be monitoring after commercialization what happens, and so that whenever there is new sightings or new developments, then they can easily adjust the law or the regulation. And so we don't really need to stick to it. If there are changes, then we change it, but it's always science-based. So if there are excesses, then that's good, but we have to look at it again, the validity of that, what is its impact, and is it reproducible? What are the conditions that made it that way? And if it's really very beneficial, then it's going to be published and let everybody knows. And that way, everybody will have to adjust or make guideline changes if needed.
Okay, thank you, Ma'am Ola. Just a quick clarification. In biotech crops, we can have multi-stack traits. There is a question about this earlier. So for example, golden rice can have a trait that is that prevents it to have a disease, right? So we have that. Another question here is, do we have gene-edited product in the Philippines, in the Philippine market already?
Yes, we have this, as I mentioned a while ago, we have this reduced browning banana, which has been approved and deemed as non-GMO. So it's already approved for commercialization and for farmers to plant if the guidelines for it is already set.
Okay, so unfortunately, that is all the time that we have. Thank you for indulging me to extend a bit. Would like to express our gratitude to our resource persons, Dr. Jones from the UK. We have Dr. Ola and Dr. Claro. I'll now turn over the floor to our moderator, Sunny. Thank you again for your wonderful questions, both here and online. Thank you very much. Sunny?
Thank you very much, everyone. And thank you very much to our participants, both online and on-site, for being very active, for those proactive questions, and of course to our speakers. At this point, we'd like to hear from the rest of the participants who may not be able to ask questions, but would like to hear some key takeaways from you. Please fill out the feedback form, and we have some questions listed in those forms. Okay. Do we still have some more prizes? Oh, we still have. So the on-site participants are very lucky to be receiving those prizes. Hopefully in the next ADSS, we'll be able to give you some more. All right, so please head to bit.ly/ADSS 15 August 2023, or you can just scan the QR code for the feedback form, and you will see some familiar questions there. So while you're at it, would like to award now the certificates of appreciation to all our resource speakers. May we invite Dr. Noor, the SEARCA Deputy Director for Programs, also Mr. Joe Florendo, SEARCA Deputy Director for Administration, and would also like to invite Mr. Mark Hansel. Okay, so this certificate of appreciation is awarded to Dr. Rhodora Aldemita for sharing her expertise as speaker on breeding innovations for increasing productivity and climate resilience in crops and livestock during the ADSS held on 15 August 2023 at SEARCA Drilon Hall, Los Baños, Laguna. The same certificate is given to Dr. Claro Mingala. All right, and the same certificate is also given from the Philippines to the UK to Dr. Hugh Jones, who's joining us from the UK. Thank you very much, sir. Okay, and finally, would like to give some tokens of appreciation to our partners from USDA Foreign Agricultural Service in Manila, for Mr. Mark Hansel, and also for Ms. Florence Sevilla. May we invite both speakers to please also join us for the photo opportunity. Tinago na ni sir. Okay, now a group photo with the speakers. Okay. We hope the participants are religiously filling out the feedback form for the essay part, 5 points each. All right, so thank you very much, everyone. Now, to formally close this very interesting ADSS, we'd like to call on the Deputy Director for Administration, Associate Professor Joe Florento.
Thank you very much. USDA Foreign Agricultural Service Manila Agricultural Attaché, Mark Hansel. Our highly esteemed resource persons, Dr. Rhodora Romero Aldemita, Professor Hugh Jones, and Dr. Claro Mingala, friends, partners, and allies here with us at Drilon Hall, and the hundreds joining us through our online platforms. Magandang hapon po sa inyong lahat. This is a diverse group of individuals. I myself come from the field of accounting and finance, and yet I have a part in advocating for modern biotechnology as an important innovation that we can use, one of our options that we can use to accelerate agricultural transformation in the Philippines and beyond. We may all be different, but all of us have a valuable contribution because food security is everyone's business. It is a very special privilege for me to conclude not just today's session, but also the whole biotech outreach series in general. SEARCA has been to the Crop Biotech Center to meet with the Nueva Ecija science community, students and farmers. We met with members of the judiciary as well as those involved in policy development at the Philippine House of Representatives. Today's session with the youth and the Los Baños science community is the cherry on top of the ice cream. We would like to thank everyone who conceptualized, organized, supported, and participated in this series of our learning session. Maraming salamat. This afternoon, the outreach series coincides or concludes, but the outreach work continues. Let us continue the conversation, the learning from each other as we go on. Let us listen to each other, learn from each other, and use what we gain today to contribute to an enabling environment for biotechnology in the Philippines and beyond. Maraming salamat po. Mabuhay po kayong lahat. Thank you.
Thank you very much, Sir Joe. So the outreach continues, but before we continue, we have some refreshments for everyone. So before you head home, we have some snacks available outside, so please grab some. But before you can grab some snacks, we'd like to receive those feedback forms. So before you leave, please make sure that you're able to fill out the jot form, okay? So that concludes our ADSS. Maraming salamat po, and until we see you again in the next ADSS. Thank you.
As an advocate ng biotech na hindi ako nahihiyang i-share yung aking experience at ako ay natutuwa na ma-share yung aking experience, kailangan natin talaga na ma-involve ang agriculture sa technology. Kung wala tayong technology, maiiwan po ang Pilipinas. Ako po si Rosalie Eliasus, taga Barangay Santa Maria, San Jacinto, Pangasinan. Isa akong farmer ng rice and corn, pero at the same time, isa akong government employee bilang Municipal Disaster Risk Reduction ng LGU San Jacinto and at the same time, the OIC SB Secretary. Ako po ay isang medical technologist grad actually. But because after na nag-OFW, umuwi ako ng Pilipinas at hindi ko alam yung gagawin ko. But bilang IPM FFS student ng government, doon ko napag-aralan ang pagsasaka both on rice and corn. Then it was in the year 2002 nung ma-involve ako sa corn IPM FFS. Pagka-graduate ko doon sa conventional corn, nagkaroon po ng field trial sa Santa Maria, Pangasinan at doon ko nakita at naobserbahan yung pagdating ng BT corn dito sa Pangasinan. So nakita namin yung mga members ng Farmers Federation ng San Jacinto, talagang nakita namin ang pagkakaiba ng BT and non-BT. At timing na 2003, maaaprubahan na pala ang BT corn dito sa Pilipinas. And from that on, not only because I started BT corn dito sa field na ito, but at the same time as the pioneer and the role model ng BT corn. Pagka conventional ka, meron talaga yung timing from the time na magplaplowing ka na maraming ilang beses. And then not only on the preparation but time of fertilization, time kung kailan ka magwi-weeding, time kung kailan ka noon ang uso sa amin is detasseling o kaya mag-i-spray ka kasi yun yung pinag-aaralan mo sa IPM FFS. With that, pati yung kung kailan ka magpapatubig, marami. At sa kung kailan ka mag-weeding, kasi maraming weeds na tumutubo. Kailangan mo pa rin na mag-uupa ka ng mga tao. So it's laborious. But with the BT corn, parang shortcut ang nangyari. Pag nakapagtanim ka na kaagad ng corn, maghintay ka lang ng at least 20 days. After 20 days, because we have 2 traits now, nag-i-spray na lang kami ng glyphosate. O di wala ng weeds at that time. Bumababa ang iyong production cost. At the same time, ang maganda is naging high yield ang BT corn. Hindi na rin nare-reject ng mga feed millers, yung mga traders. Kasi ang problema din noon ng conventional, nagkakaroon kasi ng aflatoxin. But this time, because there is no infestation, kaya wala siyang aflatoxin. So, yun yung mga differences. At pag high yield ka, di mas maganda ang profit mo. Nakita ng mga group ng farmers dito sa San Jacinto. Kaya agad-agad din silang naging adopter ng ating BT corn. And on that time, marami akong nahikayat kasi naikwento ko rin sa ating mga iba-ibang meetings, naiimbitahan ako sa mga ibang lugar na nagmamais. So, doon nag-start na parang, kaya nga nabansagan ako ng Queen of BT Corn kasi ako yung nauna na naglalaganap sa mga iba-ibang towns dito.
Ako po si Sosimo Gonzales, 70 years old, taga Kasibong, San Jacinto, Pangasinan po. Sa pagtatanim po ng BT corn, ay ang nag-introduce po sa amin yan ay sila Madam Rosalie. Tapos, nung masubukan namin, maganda naman po ang naging resulta. High yielding yung